Hello, everyone. My name is Heriberto Torres. The conference that I will be presenting today is entitled Seasonal Effect on the Anti-Proliferative and Anti-Inflammatory Activities of Bursera microphylla. To get into context, pain and inflammation are the most common and main symptoms of many diseases. Inflammation is an essential protective response to the host against internal and external environmental stimuli. When tissue homeostasis is perturbed, macrophages provide the host first line of defense against common microorganisms and bacterial infections. Toll-like receptors, macrophages can recognize LPS. In response to LPS, macrophages release soluble pro-inflammatory mediators such as cytokines and nitric oxide. Under certain conditions, the inflammatory response can become chronic and is accompanied by the overproduction of these inflammatory cytokines, which cause cellular damage. This leads to the generation of several diseases such as diabetes and cancer. The carcinogenesis process is affected in all stages, initiation, progression, and metastasis by chronic inflammation. According to the epidemiological data, 20 to 25 of all cancers are related to the chronic inflammation. Despite the progress generated in the knowledge of cancer biology and its treatment, cancer remains a leading cause of death in the world. Owing its efficacy in reducing pain and inflammation, non-steroidal anti-inflammatory drugs and corticosteroids are among the most popularly used for these conditions. However, many side effects that can be mild or serious are more appearing when these drugs are used at the higher doses of for extended periods. On the other hand, antineoplastic drugs are not selective and the cancer cell develop mechanisms of resistance against these agents. Therefore, the search for new anti-inflammatory and anti-cancer agents that are safer and more effective remain important. In this context, many plant metabolites are recognized for their therapeutic effect on various types of cancer. This is associated with their capacity to inhibit signaling pathways associated with the control of cancer and inflammation. For these reasons, plants represent a good alternative for the search for new chemical entities with anti-inflammatory and anti-cancer effects. Bursera microphylla is a medicinal plant widely used for medicinal purpose by the tribes of the northwestern of Mexico to empirically treat infection, inflammation, and cancer. In our research group, we have previously investigated the microphylla resin, showing that it has diterpenoids and triterpenoids compounds with anti-proliferative effects. In addition, we have shown that the leaves, fruits, and stems of these plants have a high antioxidant potential, which is related to the phenolic compounds. On the other hand, it is known that plants vary their profile and concentration of the bioactive metabolites depending on the biotic and abiotic factors to which they are exposed. These factors can present significant variation during the year, which directly impact the production of secondary metabolites of plants and therefore determines the biological potential that they may exhibit. In this sense, the study of the effect that the different stations have on the chemical composition and the biological properties of plants can contribute to the optimization of the parameters for the generation of herbal pharmaceutical products and to scale up. Due to the above, and because the anti-proliferative and anti-inflammatory effect of leaves, fruit, and stems of microphylla has not been studied, and also the effect of temporality on this biological activity is unknown. The main objective of this research was establish the seasonal effect on the anti-proliferative and the anti-inflammatory activities and the bioactive compound profile. To meet this objective, the following methodology was used. The specimens from each of the seasons of the year were collected between 2019 and 2020 in Caborca, Sonora, Mexico, and botanical identification was carried out. Subsequently, roots, leaves, and stems were grounded and extracted with ethanol to generate ethanolic extracts. The cell lines used in the biological assays were cultured in the medium supplemented with fetal bovine serum and maintained under standard conditions. Before the evaluation of the anti-inflammatory effect, the cytotoxic effect on the macrophage cell lines was evaluated by MTT assay. Likewise, the antiproliferative capacity of the ethanol extracts against the cell lines was also assayed by MTT. The antiproliferative activity was determined and expressed as AC50 values. In addition, the level of cytotoxicity of the extracts on normal cells was evaluated by determining the selective index. The determination of the anti-inflammatory effect of the extracts of B microphylla was carried out through the quantification of nitric oxide using the GRIS method. For a static analysis of all experiments, 2K test was used. Remarkable, all the extracts except the spring extracts of fruits and stems show no cytotoxic effect in macrophage cells. Viability was always above 90%. The inflammatory response shares various molecular targets and signaled pathways with the carcinogenesis process. Therefore, molecules with a modulation capacity of these mechanisms are candidates for the treatment or prevention of inflammatory disease and cancer. Determine the inflammatory capacity of B microphylla. If we evaluate the effect of the extracts on the production of nitric oxide. Interestingly, spring extracts were the most effective in reducing the production of nitric oxide, followed by summer, autumn, and winter extracts. In all cases, a dose response effect was observed. In general, fruit extracts were the most effective in reducing nitric oxide production, followed by stem and leaves, this according to AC50 values. In our research group, we have also tested the effect on nitric oxide production of other Mexican medicinal plants, such as Iberville sonore, which, according to previously reported, presents a graded effect compared to B microphylla. Interacting with TLR4, bacterial LPS induced the activation of several pathways. However, in future study, it is important to determine the mechanism of action through of which the bioactive compounds of B microphylla reduce the production of nitric oxide. The evaluation of the antiproliferative effect allowed to demonstrate that the cervical cancer C33A was the most sensitive to the treatment, being able to observe for all cases a reduction of more than 50% of cellular proliferation in all concentrations evaluated. In general, spring extracts were the most effective, followed by summer, autumn, and winter. The same behavior was observed in the lung cancer cells, where spring extracts were the most effective in reducing the cell proliferation, followed by summer, autumn, and winter extracts. In general, for all the evaluated doses of the different treatments, the proliferation percent was found to be closer to 50%. HeLa cells also show high sensitivity to the treatments. Of the extracts evaluated so far, summer extracts are the most effective, followed by autumn and winter extracts. On the other hand, the non-cancerous cell lines was the most resistant to the treatments. In this case, the spring extracts was the most effective, followed by summer, autumn, and winter extracts. According to the established for the National Cancer Institute, extracts that present AC50 of less than 30 micrograms per milliliter can be considered cytotoxic against these cell lines. Based on the above, all the extracts can be considered cytotoxic against the cancer cell lines evaluated. It was also observed that the non-cancerous cell lines was more resistant to the treatment. According to the AC values, all cancerous cells were inhibited selectively by the extract. At the morphological levels, after the treatment, C33A cells with the extracts was observed dead cells and cell debris, which is associated with a high cytotoxic induced by this extract in this cancer cell. In lung cancer cells, was observed cells elongate and cells shrinkage and rounding also was observed after 20 hours of treatment. This suggests that the extracts induce antiproliferative through the mechanism related to the inhibition of cell cycle and the alteration of the dynamic of the cytoskeleton. In HeLa cells, the treatment drastically modifies cell morphology, inducing shrinkage, cell rounding, and the formation of membranous webs and apoptotic bodies. This morphological change suggests that the drugs induce anti-proliferative effects through the apoptosis cell death. However, it is still necessary to explore the mechanisms of action involved and determine which are the bioactive compounds that are related to the anti-proliferative effect of microphylla. In conclusion, seasonality affect the anti-proliferative and anti-inflammatory activity of B microphylla. However, it is necessary to study the chemical composition of the drugs. Many thanks to the research group for the contribution to this study. Thank you.